Hey, Bio30s, it's Manchak here with another video lesson for you for your course. And today we are going to be covering concept C6 and concept C7. So I'm going to move that out of the way and we'll just have a quick look at what those are going to be. So, really what we're getting into, these are going to be much shorter lessons than what we covered last week with concept C4 and C5, which was a slog, and I again apologize for you for that. What we're going to work on today are some thing called sex-linked traits and understanding what those are. And it's going to work off of that sort of dominant recessive kind of idea that we were talking about so far, but we're going to dig into it in a little bit slightly different way. The other thing that we're going to talk about as well is Morgan before we get into that, and who Morgan was and why it was so important. And then a quick discussion about bar bodies as well. And then concept C7 deals with gene linkage and crossover frequency. And we'll understand what those are and move our way through that as well. You guys will be using pages 598 through 619 of your textbook. And there are 3 videos I've selected for you that I think you should honestly watch all of them. But if you watch just 2 of them, watch these 2 right here. And then if you have time, get to watching the recombination and gene mapping stuff as well. You'll have some practice questions out of your textbook, a worksheet that actually is a good review of C5 and works into C6 and C7, a practice quiz as we have done already, and a summative quiz at the end. So without further ado, I think I'm going to drag over here our concept checklist, just so you guys are aware of what we're covering in this concept. So we are covering C6 is compare the pattern of inheritance produced by genes on the sex chromosomes to those on the autosomes. And let's just take a moment. Remember, autosomes are the first in humans, the first 22 pairs of chromosomes, and then the sex chromosomes are the 23rd pair. So autosomes are the first 22. We'll get that right eventually, and then pair 23 is the sex chromosomes, and we'll talk about Morgan and a couple others in that as well. Then concept C7 is going to talk about gene linkage, crossing over, and the effects of gene linkage and crossing over on the variability of individuals. And we'll explain the relationship between variability and the number of genes controlling a trait. And we'll talk about the idea of chromosome mapping and how chromosome mapping works. So I'm going to switch over to our PowerPoint. I'm going to shrink myself down a little bit, and we are going to head into the lesson itself. So chromosomes and genetics. We're going to start out with just a quick, sort of where are we at right now, and sort of fill in the blanks, and then we'll move from there. Remember, we've covered in this section of Unit C so far a quick introduction to Mendelian genetics, and then we looked in C5 at a whole bunch of different patterns of inheritance. We looked at things like monohybrid crosses and the patterns that are associated with that. We looked at codominance, incomplete dominance, multiple alleles, and the patterns associated with that. We looked at dihybrid crosses, and again, the patterns associated with that. And we also took the time to look at some pedigrees and try to understand what those are. What we're feeding into now is another pattern of inheritance, right? We're looking at sex-linked characteristics that are linked to the 2 sex chromosomes that we have in humans and many other organisms. And so that's what we're going to head into now. So just a review, just to sort of remind ourselves what's going on here. Humans have 46 chromosomes in their body cells, and I want to specify their body cells, their autosomes, because sex cells are a little bit different. Okay, so there's 46 chromosomes comprised of 2 sex chromosomes and 44 autosomes. Okay, the sex chromosomes determine our sex. So we know that if you have 2 X chromosomes, you're female. If you have 2 an X and a Y chromosome, you are male. And there's other characteristics that are tied into those sex chromosomes as well. And we're going to explore a few of those as we move forward. So autosomal chromosomes, or autosomal disorders, or something that is autosomal, is referring to anything that occurs on the 22 pairs of chromosomes or the 44 autosomes, and not the sex chromosomes. Okay, So they would be non-sex-linked genetic material. Genes that are located on the same chromosome, just as a definition, are called linked genes. And what that means is maybe we've got a chromosome that determines this trait, and we got our chromosome here, and we got a gene on the chromosome up here that determines one trait, and we got a gene down here that determines another. Actually, we got a whole bunch of genes in between there. And all of those genes are considered to be linked genes because they are on that same chromosome. Okay, so just a bit of a set the stage for us. Where we're moving into now is starting to, again, find some patterns that don't quite make sense based on what we saw from Mendel and his genetics. And so we certain scientists have gone and explored that a little bit more deeply. So the chromosomal theory of inheritance. Early research into chromosomal theory involved looking at chromosomes and how they segregated during meiosis. And it was observed during this process that chromosome pairs separated and then reformed when sperm and egg united. And we know this, right? We have segregation, independent assortment, all of that stuff happens during meiosis where we line up along the metaphase plate, and those pairs of chromosomes, one pair from your mother, one pair from your father, get separated. That tetrad gets separated, but it could be mother, father, or it could be father, mother, right? Depending on the situation. It was also observed by Mendel that factors from both egg and sperm contributed to characteristics on offspring. So Mendel, remember I told you that we didn't have at that time a sense as to what carried the genetic information. We knew the genetic information was there, and he knew of this thing called heredity, but he really had no idea what carried it. So he called them factors. Didn't really know what it was. Eventually, it was determined chromosomes carried the factors or alleles that coded for these characteristics. Okay, and those alleles and how they work together are called genes. Those genes then are the specific set of instructions to make a particular characteristic, a particular protein, as we'll see in the next part of the unit. This led then to the development of the chromosomal theory of inheritance. And this is what it says. It says that chromosomes carry genes, and they are the units of heredity. And that paired chromosomes segregate and independently assort during meiosis, with each gamete having half the number of chromosomes found in somatic cells. Somatic cells, remember, are your body cells. Sex cells are your sperm and egg. Sex cells, remember, are N. And your body cells are 2N. So there's those words, segregation and independent assortment, again, that are cropping up again and again as we move through this section of this unit. Okay, so the chromosomal theory of inheritance is that chromosomes carry genes, which are the units of heredity, and that paired chromosomes segregate during meiosis after they've independently assorted during metaphase 1. And then when we've finished meiosis and we have gametes, those gametes have half the number of chromosomes found in somatic cells. And we know then that when one gamete and another gamete come together, sperm and egg, then we have a reuniting or creation of those pairs of chromosomes again that were segregated but now are brought back together. So now we get into talking about Morgan and what Morgan did to explain sex-linked characteristics. And it's kind of similar to what we saw in dihybrid crosses. And it's all really about evidence at this point. He went and did experiments with Drosophila, again, our fruit flies. And he started to play around with it. And he got expected results, and then he got unexpected results. And remember, in science, it's those unexpected results that often lead to the most important discoveries that we come up with. If we knew exactly how everything was going to happen, science would be pretty boring as far as I'm concerned. So a bit of background, and then we'll jump into the expected and the unexpected that Morgan encountered. He used fruit flies because, again, the fruit fly reproduces rapidly. The offspring are capable of mating shortly after hatching, and females will produce over 100 eggs after each mating. So all of that tells you right away that we can produce lots of generations relatively quickly and get a very large sample size. And large sample size is really important when you're doing science, because then it adds validity to the research that you're doing. Many flies can also be housed in the same test tube, which means that it's a small experiment, not a giant. If you're trying to do the same thing with mice or something like that, you'd need a huge lab to do it. You can do this with a small lab. And males can be easily distinguished from females. Males have a small dark abdomen, while females have a large banded abdomen. So really easy to separate males and females, really easy to see some of the characteristics under just a simple dissecting microscope. So with that background, just a little bit more here. Fruit flies have 8 chromosomes, so they have 6 autosomes and 2 sex chromosomes, and they have an XX and an XY, just like humans do. And so here's what Morgan started to notice, okay, and where things went a little bit wonky. Morgan noticed a white-eyed male in his group of flies. He crossed with a male red-eyed female, and that made absolutely no sense. Let's try that again. Morgan noticed a white-eyed male in his group of flies. He crossed that male with a red-eyed female, and all the offspring were red-eyed. That's pretty expected because red is dominant to white, and so he expected that that was going to happen. Then what he did, though, is he crossed 2 of the hybrid eyes from above and noticed that 1/4 of the new offspring were white and 3/4 were red-eyed. That's normal. That's explained by Mendelian genetics, right? Because basically what he's done is created these offspring that were red-eyed. I'm going to get my pen out just so you guys can visualize what we're doing. What color do we want? Let's do yellow. These offspring here who were red-eyed, they would have been, I'm just going to put R's because they're easy to distinguish and relatively easy to write. They were all going to be heterozygous when they're done. And so if we took 2 of these and we crossed them, and you guys can do this Punnett square on the side if you like, but we're going to end up with a 3-to-1 ratio. 3/4 are going to be red-eyed, 1/4 are going to be white-eyed. Okay, that's pretty logical. But here's where things got a little wonky for him. On closer examination, he noticed that none of the white-eyed individuals were female. Now, the way things normally work is that there should be a 50-50 ratio if we were not dealing with an unusual situation. In other words, of those white-eyed individuals, 50% of them should have been male, 50% of them should have been female. None of them were female. That was odd. And Morgan wasn't sure what was going on with that. So out of all of the males in F2, so this is the second filial generation, 50% had red eyes and 50% had white eyes of just the males. Okay, that was weird. Shouldn't have been that way. And so this led Morgan to think there's got to be some other pattern of inheritance going on here. And he figured eventually, after doing some work, that there must have been something going on with the difference between males and females. This video right here, I would encourage you to hit pause quickly. It's only a 3-minute and 30-second video that just quickly summarizes the experiment as well with a couple more visuals just to help you out if you want a little more reinforcement. So hit pause now if you want to do that, and I will move on. So here's what happened. After Morgan did his experiment, he started to do some examination. So he noticed then that the 2 sex chromosomes in the male were not the same size and shape as they were in the female. Okay, and so that led him to believe that something was odd, right? So here are the sex chromosomes. I'll switch to a different color so you guys can see a little bit better. So here's the sex chromosomes in the male, and here's the sex chromosomes in the female. Okay, so these two are identical. Those are your 2 X's, and then these 2 are not identical. There's an X and a Y. So that was unusual. Morgan determined then that eye color gene was located on the X chromosome. So it was located on this chromosome and on these 2 chromosomes, okay, depending on if you're male or female. Since males only have 1 X chromosome, if they received 1 recessive allele, that they will show that trait because they don't need the other recessive allele because it can't be on the Y chromosome. In order for females to be white-eyed, they must have 2 copies of the white-eyed gene. That's why he didn't see any females come up in that experiment, because the male was already red-eyed. And the female that he brought in was red-eyed as well, but must have been heterozygous. Okay, so I'm going to show you what that looks like in just a second. In order for, so Morgan had just discovered that traits that are carried on the sex chromosomes are what we call sex-linked traits, and that's what he was able to discover, is that there are genes that are carried by the X and the Y chromosomes, particularly the X chromosomes. So what I would like you guys to do right now, because I think it's going to be better to explain it, is go and watch this video, please, on sex-linked traits. Hit pause, watch the video, and then come back to me afterwards. And this does an excellent job of explaining not only what sex-linked traits are, but also how we do the Punnett squares. And pay close attention, please, that we're going to use some alternate notation. So if you have a male and a female, okay, the female will be XX, the male will be XY, and that will put superscripts and subscripts, sorry, superscripts up here to represent the alleles that the X chromosome is carrying. Okay, and this would actually incidentally have been the cross for what we saw up here in this cross here. Okay, so that's what we ended up with. Okay, so go watch that video now, and I'll meet you back here in just a second. So in the video, what ended up being covered was this sort of cross. So this is looking at the generations and what ended up happening. And I'm just gonna scroll back up and remind you what went on up here. So Morgan noticed after he did a cross that he had a white-eyed male in his group of flies, and he crossed that male with a red-eyed female. So I'm going to jump ahead. Here's your red-eyed female. Here's your white-eyed male. Okay. What ended up happening is all of the offspring came up being red-eyed, which was to be expected because we had a situation of being overruled here. The big R overruling the little r. But then what happened and where it got really weird is that we ended up with a situation where he took the females that came out of this group. So he took that female, for example, and this male, for example. And what he expected to happen is they should all come out red-eyed. He was pretty sure. Or we should get a 1-to-3 ratio. And he did. He got a 1 white-eyed to 3 red-eyed. But the problem is that when he looked at male versus female, he saw that none of the females, and these are the females here, were at all, or were white-eyed. And that 50% of the males, so 1 and 2, happened to be white-eyed. So this points to this kind of pattern of inheritance. And notice the notation that you have on this diagram where we've got X big R Y, X big R, X little r, and how these are females up here and that these are males down here. And that for a male to express the recessive allele, to express the recessive trait, they only need one of those alleles to express it. And that's the big trick about sex-linked characteristics. Okay, we'll often talk about color blindness with sex-linked characteristics as well. So sex-linked traits are controlled, come on, little pointer. There we go. Sex-linked traits are controlled by genes located on the sex chromosomes. A recessive trait located on the X chromosome is more likely to express itself in males than females. This is because males need only one copy of the recessive allele. Looking at the numbers, this also means that our probabilities and proportions will be different than what Mendel's proportions would be if we look only phenotypically. Okay, so again, as we've gone through all of these different patterns of inheritance, what we've noticed is that when we shifted away from the typical Mendelian genetics and what he came up with, our patterns start to get thrown off. Our probabilities aren't there. Phenotype ratios aren't showing up the way they should be, and so we end up with different results than what we'd expected. All right, so moving on, if I can. There we go. What I wanted to talk briefly about here are the human sex chromosomes. Move away from the Drosophila now really quickly. In humans, the X and Y chromosome, the 23rd pair of chromosome, contains a small region that is homologous and a larger region which contains different genes. So this is our homologous region here, You can notice it's represented by the same length and color. And then these are our differential or different regions. Okay. Some examples of human sex-linked traits include color blindness, where more males exhibit color blindness than females. There is a myth out there that you cannot be colorblind if you're female. It is possible, but you have to have a mother that carries the color blindness gene and a father who is colorblind in order for that to happen. Also, infantile spinal muscular atrophy, which we find more males. We call these X-linked traits or sex-linked traits. If only males show the trait, then the gene is carried on the Y chromosome. So, for example, having hairy ears, that's only something that happens to males because it is carried on the Y chromosome. Just a quick note about something called Barr bodies. Dr. Murray Barr noticed a dark spot in the nucleus of some somatic cells of female mammals during interphase of meiosis. This has become known as a Barr body. It was identified as sex chromatin, which occurs when one of the X chromosomes in the females randomly becomes inactive in the cell. So from this, it was determined that not all female cells are identical. Some express one and not the other X chromosome, and some express both, even though they're genetically identical. So what this did is it led to, if you imagine you're heterozygous, and maybe in one of the cells, the dominant the dominant allele had been shut down and not expressed for whatever reason, then you're going to express the recessive allele when you normally wouldn't have expressed this recessive allele. So this led to discovery of disorders like anhydrotic ectodermal dysplasia, which is a skin disorder where females have patches of skin that contain sweat glands and patches that do not, right? And we can say that the patches do not, maybe that's recessive, and maybe that's where those cells have shut down the dominant allele, and only the recessive allele is being expressed if that female is heterozygous. So at this point, I'm going to ask you guys, you can pause and do these questions on page 634. I would encourage you also to have a look at the hemophilia information on page 632 and maybe just read through it. There's a nice big pedigree on there that might be worthwhile having a look at if you're not comfortable with hemophilia stuff, and if you're not comfortable with pedigrees. Oh boy, that's bad, because hemophilia and pedigrees, and I almost mixed it too. Okay, anyway, we'll carry on. That's the end of concept C6. So if you guys wanted to pause and take a break for a bit, you can do that. I'm going to jump right into concept C7, and we'll see you over there. So concept C7 really deals with two things: gene linkage and crossover. And we said back at the beginning of concept C6 that gene linkage is we would describe linked genes as genes that are found on the same chromosome. Okay, and what this leads us to is that it leads us again to patterns of inheritance, those phenotypic ratios that just don't make sense for whatever reason. So we'll fill out a table here, and we'll work with some numbers again here. So again, we're going back to Morgan and some of his experiments with all of this stuff. So Morgan noticed some unexpected combinations of genes during his experiments. The expected phenotypic ratios were not right when he actually conducted crosses. That led him to sex-linked characteristics. It also led him to his ideas of gene linkage. When Morgan tried crossing two flies heterozygous for body color and wing shape, he expected to see this cross. Now, I'm only going to fill in the beginning. You guys can fill in the blanks on this if you'd like to. It's simply a dihybrid cross. So each individual, we're going to select their gametes. Okay. So we're going to be doing the gametes as we would have expected to be. Sorry, that should have been. Do I have an eraser tool on here? I know I do. Give me my eraser tool. Okay. We know what we can expect from this. So this, if you recall, if you've done your work over on concept C5 on your dihybrid crosses, you know that both of these individuals are heterozygous for both traits. And what that means then is we are going to produce, and I'll write it in a different color here, we are going to produce a 9 to 3 to 3 to 1 ratio. Okay, so that 9 to 3 to 3 to 1 ratio is what should have been produced in these individuals. Okay, fill that in if you'd like to. Okay, I'm going to move on. Instead, almost all the flies with wild type body had straight wings, and all that had black body had curved wings. Okay, meaning that instead of independent assortment, these alleles were staying together during meiosis. So what this led Morgan to believe is that instead of the wing shape and the body color being on two separate chromosomes, they were actually on the same chromosome. What that meant is the chances of them separating were very, very, very small. Okay, let me highlight that again. So we would have expected a nice 9 to 3 to 3 to 1 ratio. But what ended up happening is we ended up with the wild type body and the wild type body color with straight wings, and then the other ones had black body and curved wings paired together at higher proportions. We expected something different, we got something else. What this led Morgan to believe is that these two chromosomes were on the same, or these two alleles were on the same chromosome, meaning that it was difficult for them to be separated during meiosis. If they were on separate chromosomes, there would have been randomness assigned to that, and we would have got what we expected. So when Morgan was doing his crosses, he also noticed a very small amount of the flies from the original cross were black body straight wings or wild type curved wings. I'm going to show you the next slide and I'll try to explain this. How is this possible? Okay, if the genes were supposed to be linked to each other and therefore had to move in the same direction during anaphase of meiosis. What we're saying is, if you remember, when we line up, we independently assort, and those pairs in meiosis get separated from each other in the first separation. Well, if they get separated from each other, but they're on the same gene or on the same chromosome, they're really not being separated from each other. So we would expect that we would end up with only wild type straight, black body curved, right? Those combination, having wild type and straight, having black body and curved. We would expect only those combinations. But what he found is that there were some that were black and straight, black body, straight wings, and there were some that were wild type and curved wings. Okay, that was unusual. He did not expect that. If they were supposed to be on the same chromosome, we ended up with other options. What ended up happening? Okay, and I'm going to skip ahead to this slide to sort of, or no, I'm not. What ended up happening and what we ended up discovering is that there is crossover that takes place. Remember, in prophase 1, when those tetrads start to get close to each other, and start to pair up, what ends up happening is they swap, they cross over, and they swap some genetic material, coming back just a little bit different from where they started. So that during crossover and meiosis, which doesn't always happen, but does happen infrequently, genes are exchanged between homologous chromosomes. So that's what we're showing over here. I'm getting my highlighter out. So we've got this individual and this individual, and we end up with small amounts of variation that are taking place in those individuals. So in rare cases, genes that were supposed to stay together during meiosis ended up switching due to crossing over. And so that would explain us getting these genotypes from the individuals, even though if they were on the same gene, they should be linked and we wouldn't get any of that separation taking place. So Morgan's experiment showed that this crossover always occurred with the same frequency, what we call crossover frequency. And it's represented by a percentage and can be calculated using the number of recombinations over the total number of offspring multiplying by 100%. The number of recombinations refers to the number of flies produced that were not expected in the cross. So let me get to this next diagram here on this page. So based on the information below, we have a crossover frequency of, these are the ones that weren't expected here. So there was 9 in his, I think it's 400-ish individuals that he had not expected to come over if the genes were linked. And so that was a crossover frequency of about 4.5%, right? 9 plus 9 is 18, divided by 400 total individuals gives you 4.5%. And what this is showing is that we get that this is what we would expected to happen if the genes were linked together, but we ended up with a small proportion, right? We should have ended up with just these. So just this phenotypic expression and just this phenotypic expression, but we had a tiny amount of those. And the tiny amount of these unusual ones was explained by a tiny amount. In 4.5% of the individuals, crossing over did occur, leading to a tiny amount of unexpected phenotypes. I hope that makes sense. I don't know if I did the best job explaining that to you. Luckily, we've got a video coming up on this as well. So what I also wanted to talk about then is we can sort of get an expectation as to how frequently crossing over will take place based on how close those two linked genes are on the chromosome. Okay, very, very simply, the way to think about this is if one of your linked genes is way at the top of the chromosome and the other is way at the bottom, there's a really good chance, a much better chance that we could end up with a cut anywhere along the way here that would cause them to be separated. The closer they are together is a much smaller chance of them actually being separated from each other, right? If your chromosome is this big, but your genes are that close together, you've only got this much of your giant chromosome possibility of separation. So high crossover frequency if they're very far apart, low crossover frequency if they're very close together. So knowing this allows us to predict where on the chromosome a particular gene is in relation to other chromosomes. What this is allowing us to do is map a gene and figure out map a chromosome and figure out what genes are on that chromosome and where they might be on that chromosome. Is it near the top? Is it near the bottom? Is it somewhere in the middle? How close is it to other ones? Okay. So for example, two genes that have a crossover frequency of 1% are much closer to each other than two genes with a crossover frequency of 12%. So this is what we would do to map these genes. We'd look at crossover frequencies, and that's what these percentages are. And we would, in this case, we're using map units. And these are our two possible alleles, right? But what we'll see is that these two are very close together. There's only 13 units difference between them. So it would be much less chance of these two experiencing crossover than, let's say, the long feelers and the red eyes. They're very far apart on this chromosome. So there's a good chance that we're going to end up getting a cut somewhere in between here, causing those two to be separated and ending up with a different phenotype. So here's how we would work with one of these questions. And play around with it a little bit. And I'm just going to simply do some really poor drawing, but hopefully we can figure it out together. So I'm going to move this over here. From crosses between different Drosophila, a geneticist finds that the crossover frequency between A and B, okay, so A and B is 12%. Okay, so then the crossover frequency between B and C is 7%. So just from that information, there's a much greater chance of A and B being split up than B and C being split up. And then the difference between A and C is now 5%. Okay, so what this can do is asking us what is the order and relative distances of these three genes on the chromosome. So I'm going to draw a really poorly drawn chromosome, okay, and I'm going to look at these values. And I'm going to try to say to myself, okay, where would these chromosomes possibly be? I'm going to employ a little bit of logic here. This is the biggest number right here. So that means they're the furthest apart on the chromosomes. So we might say that that gene A is located at one end and gene B is located at the other end. Okay? And that this here is a 12% crossover frequency chance. Does that make sense? They're the furthest apart on there. Now, that means then that C has to be going somewhere in between A and B. But we can do some logical deduction here. Is C going to be closer to B or A? Well, if there's 12% difference here, that must mean that C is going to be slightly closer to A than it is going to be B, right? If we imagine this is the midway point here, okay, then C's got to be a little closer to A than B, because this has to be 7%. And this is going to have to be 5%, because 5 plus 7 gives me 12. Okay, so when we're mapping the chromosomes using chromosome frequency, all you're going to do is take your biggest number, your biggest crossover frequency, and put those at the opposite end, and then try to space in between. So really you're playing a logic puzzle to try to make these make sense for you. Hope that does make sense a little bit. I included, I'm just going to go back into here. This, I didn't include the video in here, but this video on genetic recombination and gene mapping, please watch it if that stuff didn't make sense to you, with Morgan's experiments and the linked genes and all of that kind of stuff. Please give that a listen and a watch to make sure that you're comfortable with it. So you can jump into practice right now on page 641 to get a little bit of work on this. And then I'm going to just go back to this slide one last time and just quickly talk to you about what else you can do. So if you haven't watched these videos, please do them. Do watch the genetic recombination video. I think it's really quite good and be worthwhile you guys having a look at. There's your practice work. Do concepts, or do worksheet 5, please. It's not terribly long. You can skip over some of the stuff at the beginning if you want to, if you feel comfortable with C5, and then really just skip to the C6 and C7 stuff. There's some diploma questions at the end that'd be worthwhile looking out. Do your formative quiz, and then as always, guys, please make sure that you wait for me to give you feedback before you move on to the summative quiz. If you have any questions, if things didn't make any sense, please reach out to me by email or pop in for my office hour anytime, and we can discuss things and sort things out. Stay caught up with this stuff, otherwise you end up way too far behind and it's tricky to get yourself caught up. So as always, stay healthy, and we will talk soon when we get into concepts C8 and C9.